morning everyone, respected teachers, my dear learned colleagues. I am Deepak Vishwas present before you to give a seminar on the topic role of vitamins as coenzyme. First, as we start, we need to know what are vitamins and coenzyme. Vitamins are organic compounds required in small amount in our daily diet, which are not synthesized in adequate amount in our body, and we need that from our diet or supplements. Usually classified into two types, water soluble and fat soluble. In water soluble we have vitamin B and vitamin C, and in fat soluble we have vitamin A, D, E, K. And coenzymes are organic non-proteinaceous organic molecules which are attached to the main enzyme at the active site and help the enzyme in the coenzymatic activities function. And how do vitamin acts as coenzyme is the main question we are going to answer in this seminar. So first the vitamin is ingested in our body, and after that its conversion into active form. Then the binding of the enzyme, the binding of the coenzyme to the enzyme, which is the crucial step, and after which the enzyme functions, and even the coenzyme can be reused after being used one time. The role of coenzyme in our body are mainly in amino acid metabolism, DNA synthesis, etc. For further details, I would like to call Dipjit.
So to begin with the first B vitamin, vitamin B1, thiamine. Thiamine. The main source of thiamine is alluvial layer of rice. It also comes from whole grains, legumes, nuts, etc. Mainly absorbed in jejunum and ileum. The coenzyme form of thiamine is thiamine pyrophosphate. Once thiamine is absorbed in our body, it reacts with ATP to form thiamine pyrophosphate.
One second. Please try to concentrate on only coenzyme activity.
Yes, sir.
We don't need sources of vitamins here.
Functions of thiamine pyrophosphate. It is crucial for key metabolic enzymes like oxidative decarboxylation reactions of alpha keto acids like alpha keto glutarate dehydrogenase and pyruvate dehydrogenase, also in trans ketolase reactions. The main riboflavin has two active coenzyme forms, FMN flavin mononucleotide and FAD flavin adenine dinucleotide. Riboflavin reacts with ATP to form flavin mononucleotide, and then it again reacts with ATP to form flavin adenine dinucleotide. Functions of FMN and FAD: it acts as electron carriers in oxidation reduction reactions, acts as coenzyme for flavin proteins in electron transport chain of complex 1, and succinate dehydrogenase, where succinate gets converted to fumarate and FAD acts as a coenzyme. Now I would request my other friends to continue on the topic.
Vitamin B3, niacin or nicotinic acids, or it is called niacin or nicotinic acids, dietary sources found in the meat, whole grains, legumes, mushrooms, fortified cereals. It is not a true vitamins. It is partial synthesis, 60 mg tryptophan to convert it into the 1 mg niacin, required as a B6, B2 and iron as a cofactor. Absorption in small intestines through a active transport and passive diffusion. Here it is an active form of NAD plus and NADP plus. NAD plus, it is a catabolic pathway, and NADP plus, it is an anabolic pathway. Its deficiency: 4 days, diarrhea, dementia. dermatitis, and it is untreated to called as a death. Then vitamin B5 or pantothenic acids. It is daily intake 10 mg per day. Dietary sources, it is liver, egg, meat, whole grains, and absorbed in jejunum. It is primarily in coenzymes, and then converted into the free pantothenic acids. In tissue, pantothenic acids plus ATP to form, it is a 4-phosphopantothenates. Function as a CoA acetyl group, crucial TCA cycle, fatty acid oxidation and synthesis of cholesterol acetylcholine. Its deficiency, 4-phospho synthesis of cholesterol acetylcholine and vitamin 5 or pantothenic acids deficiency. It is a hotness of lead. Vitamin B5 or pyridoxine. 1 coenzymes PLP 2 mg per day is daily intake. 3 vitamins pyridoxine, pyridoxal and pyridoxamine. 4 it is the sources of plant sources and animal, absorbed in jejunum and passive diffusion. Primarily it is a PLP or pyridoxal phosphates. Conversion, all forms are converted into to active PLP in liver. Functions, amino acid metabolisms, transamination, deaminations, heme synthesis, ALA synthesis, neurotransmitter GABA, serotonin and tryptophan to the niacin conversion. Vitamin B6, then Deepsika.
Vitamin B7, absorption primarily in the small intestine via sodium-dependent active transport or passive diffusion. Avidin or raw invites inhibits absorption. Coenzyme form and function, active form, Biotin covalently is bound to lysine and carboxylase enzymes in the form of biotin. Reaction, Biotin plus ATP converts to biotinal AMP. Biotinal AMP transfers to apocarboxylase, forming holoenzyme catalyzed by holocarboxylase synthetase. Function, coenzyme for carboxylation reactions carrying the activated carbon dioxide. Key Biotin-dependent carboxylase enzymes, pyruvate is converted to oxaloacetate by pyruvate carboxylase in gluconeogenesis, and acetyl coenzyme is converted to malonyl coenzyme by acetyl coenzyme carboxylase, which is the first step in fatty acid synthesis. Next vitamin is vitamin B9, or folate or folic acid. In the left is the structure of folic acid consisting of the pteridine ring, para-aminobenzoic acid and glutamic acid. Absorption and coenzyme formation, it's mainly absorbed in the jejunum, and folate is deconjugated to its monoglutamate form. It's then reduced to tetrahydrofolate, which is its active coenzyme. Folic acid is reduced to dihydrofolic acid, which is further reduced to tetrahydrofolate, and it's with the help of 2 molecules of NADPH, and it's by the enzyme dihydrofolate reductase. Functions of THF derivatives, 5,10-methylene THF is involved in DNA synthesis, and 5-methyl THF is essential for methionine synthesis, in which homocysteine is converted to methionine with the help of vitamin B12. Moving on to vitamin B12 or cobalamin, its active coenzyme forms, it's converted to methylcobalamin for methylation reactions in cytosol and adenosylcobalamin for isomerase reactions in mitochondria. It has a complex absorption process. It's released from food by gastric acids, binds to R proteins and then to intrinsic factor in the duodenum. The B12 intrinsic factor complex is absorbed in the ileum and is transported by Cobalamin 2. And its key coenzyme functions are methylcobalamin converts homocysteine to methionine by methionine synthase, avoiding the folate trap. Adenosylcobalamin converts methylmalonyl coenzyme to succinyl coenzyme via methylmalonyl coenzyme mutase, vital for fatty acid and amino acid metabolism. The last vitamin is vitamin C or ascorbic acid. Its active form is L-ascorbic acid. It acts directly as a coenzyme or reducing agent without requiring conversion. Absorption site and mechanism, it is mainly in the small intestine and proximal jejunum. Mechanism, active transport through sodium-dependent vitamin C transporters 1 and 2 and passive diffusion at high concentrations. Absorption efficiency decreases with an increasing dose. Coenzyme reactions, vitamin C functions as a cofactor for hydroxylation and redox reactions, particularly in prenyl hydroxylase, hydroxylation of proline to hydroxyproline in collagen synthesis, and lysyl hydroxylase, hydroxylation of lysine to hydroxylysine in collagen cross-linking. And we've come to the end of the presentation. Thank you.
Well, thing is, it was a good reading show. It was not at all a seminar. You all just read what is written over there. This is how do we take classes? Is this how we take classes? Suppose you have been asked to tell about the coenzyme activity. Where are the reactions? If I ask you who taught us NAD, name one reaction, give one example where both NAD and NADP can be used. Give one example where both NAD and NADP can be used. Either of these. Answer?
Oxidation reduction reaction.
In the mic you answer.
Oxidation reduction reaction.
Give one example, specific example, and that is a very important enzyme, clinically important enzyme. It is being estimated in the laboratory. Correct? So you missed that. So what you have done, you have copied, fine, but you have not read it. You have not gone through. This is not the way you present seminar. This shows you are very ill prepared. Your preparation was not good. Slides also is not good. Whatever you can copy, but maintain some decorum. This is so small, I doubt anyone could see from the last bench. Even you can't read standing here. The font size is so small. Can you find any of my slides like this? What is this? Just write everything and go on reading. Okay? So this is a learning process. I hope in the next time you will do it better. Next group, please.
A very good morning to respected professors and my dear batchmates. Myself, Disha Sengupta, and today our team is going to present a presentation based on the topic hyperlipoproteinemia. So let's begin with the introduction. So firstly, we have what is hyperlipoproteinemia. Hyperlipoproteinemia means elevated levels of lipoproteins like cholesterol and triglycerides present in the blood. Its significance includes major risk factors for diseases like atherosclerosis, coronary artery disease, and pancreatitis. Next, we have classification of lipoproteins. Lipoproteins are classified into chylomicrons, very low density lipoprotein, low density lipoprotein, and high density lipoprotein. Moving on to types of hyperlipoproteinemia. According to the Frederickson Classification, which is World Health Organization adopted, we have type 1, where the elevated lipoproteins are chylomicrons, elevated lipid is triglyceride and associated disease is pancreatitis. Type 2A, where the elevated lipoprotein is low density lipoprotein, elevated lipid is cholesterol, and associated disease is coronary artery disease. Type 2B, where elevated lipoprotein is low density lipoprotein and very low density lipoprotein, elevated lipid is cholesterol plus triglyceride, associated disease is coronary artery disease. Type 3 the elevated lipoprotein is intermediate density lipoprotein, elevated lipid is cholesterol plus triglyceride, and associated disease is peripheral vascular disease. Type 4, elevated lipoprotein is very low density lipoprotein, elevated lipid is triglyceride, and associated disease is coronary artery disease. Type 5, the elevated lipoproteins are chylomicrons and very low density lipoprotein, elevated lipid is triglyceride plus cholesterol, and associated disease is pancreatitis. Now I would like to hand over the mic to my next team member.
Hello everyone. I'm going to continue from here on the different causes of the types of hyperlipoproteinemia. So first up we have type 1 hyperlipoproteinemia, which is also known as familial chylomicronemia syndrome. Why familial? Because it runs through families. It is inherited. It is autosomal recessive. We have a cause of genetic mutation. Either it is caused by ApoC2 deficiency or LPL deficiency. ApoC2, it basically works by activating LPL. What does LPL do? LPL hydrolyzes triacylglycerol into fatty acids and glycerol. It breaks it down into its free components. If any of these is defective, we have reduced breakdown of chylomicrons, increased triglycerides in the blood, which results in milky or cloudy plasma, also known as lactocin plasma. It can be used as a diagnostic approach for type 1 hyperlipoproteinemia. Which we have hepatosplenomegaly because of lipid deposition on liver and spleens. Eruptive xanthoma is a clinical feature which are basically raised papules on skin. We have increased risk of pancreatitis. Next up, type 2 hyperlipoproteinemia, also known as familial hypercholesterolemia. It is a mainly autosomal dominant inheritance. In type 2 hyperlipoproteinemia, we have mainly the causes of mutation in LDL receptor present on liver or ApoB100 receptor on LDL molecule. What happens basically is ApoB100 binds to LDL receptors on liver. It helps in receptor-mediated endocytosis. As a result, the liver takes up LDL. The cholesterol is converted into bile acids and it is excreted out from the body. If the mechanism is defective, we have reduced LDL clearance and increased LDL levels. In clinical features, we have tendinous xanthoma. Next up, type 3 hyperlipoproteinemia, also known as familial dis-beta lipoproteinemia. It is caused due to a genetic mutation in ApoA gene. ApoA gene works similarly by binding to ApoA receptor on liver, which helps in its receptor-mediated endocytosis and clearance. As a result of it, if it is defective, it causes delayed clearance of chylomicron remnants and VLDL remnants. VLDL remnants are also known as intermediate density lipoprotein. Therefore, there is an accumulation of remnant lipoproteins in plasma. As a result, there is increase in total cholesterol and triglycerides. In clinical features, we have palmar xanthomas, tuber eruptive xanthomas, premature coronary artery disease, and pulmonary artery disease. So these were three types of hyperlipoproteinemia for which the main causes were genetic mutations. We also have type 4 and 5 for which causes are multifactorial and not only caused due to genetic mutations, which are also influenced by lifestyle factors like alcohol intake, other health conditions like diabetes mellitus, or certain medications like steroids. Thank you. Now I'm going to pass on the mic to
তো আমরা জেনে নেব যে হাইপার লাইপোপ্রোটিনেমিয়ার কজ কি কি। প্রথম প্রাইমারি কজ আছে আর কিছু সেকেন্ডারি কজেস আছে। তো প্রাইমারি কজে প্রথমেই আমরা চলে যাব ফ্যামিলিয়াল হাইপার কোলেস্টেরল।
Sir, don't understand Bangla.
স্যার আই এম নট ইউজড টু ইন স্পিকিং।
Sir, don't understand Bangla.
So we'll first go to the familial hypercholesterolemia. Familial hypercholesterolemia is a genetic disorder where there can be mutation in two genes, LDLR receptor genes, where LDL receptor are not working correctly, and the second is PCSK9 gene, where LDL receptors are degraded. That's why familial hypercholesterolemia occurs. Secondary causes are like diabetes mellitus, hypothyroidism, alcoholism, and other drugs like thiazides and steroids. Clinical features are xanthomas. Xanthomas is abnormal swelling where yellowish cholesterol deposition occurs in the skin, tendons, or other body parts. It can be named tendon, eruptive, or tuberous in various locations on the basis of various locations. If it occurs in tendons, it called tendinous xanthomas. If it occurs in joints like knees or elbows, it called tuberous xanthomas. And if it occurs in shoulders and extensor limbs, it called eruptive xanthomas. Xanthomas is a marker where we can visibly mark lipoprotein disorders, lipid disorders. Then xanthelisma. Xanthelisma is nothing but xanthomas if it occurs in eyelids, where yellowish cholesterol deposition occurs in eyelid. Then corneal arcus. As the name suggests, arcus is arc-like or semicircular ring-like appearance which occurs in cornea. Complications are premature coronary artery diseases. Premature coronary artery diseases where arteries outside hearts are affected, where arteries are blocked or narrowed due to deposition of lipoproteins like cholesterol and triglycerides. It can occur in renal arteries where renal dysfunction can occur, or it can also occur in mesenteric arteries where postprandial abdominal pain is seen, which is also known as intestinal angina. And there is pancreatitis. Pancreatitis occur due to degradation of lipoproteins into free fatty acids, triglyceride lipoproteins, where triglyceride lipoproteins are degraded into free fatty acids, which causes ischemia and capillary blockage. Then we got cerebrovascular accidents, which is commonly known as stroke, which occurs due to absence of blood supply in the brain due to blockage of like cholesterol and triglycerides, which blocks the artery and blood supply to the brain is damaged. Then we got peripheral vascular diseases. I will hand over the mic to next person.
So the diagnosis are fasting lipid profile, total cholesterol, low density lipoprotein cholesterol, high density lipoprotein cholesterol, triglycerides, and apoproteins level. Optional secondary cause evaluations. Treatment principle: lifestyle modifications like diet, exercise, weight reduction, and avoiding alcohol. Diet constitutes of low fat and high fiber. Pharmacological treatment, drug class like statins lowers cholesterol levels and reduces the risk of heart diseases. And example, atorvastatins lowers low density lipoproteins levels. Fibrates, fenofibrates lowers triglyceride levels. Niacin, nicotinic acid increase the high density lipoproteins, that is good cholesterols, and lowers triglyceride levels. And ezetimibe lowers cholesterol absorptions. And PCSK9 inhibitors, evolocumab, lowers low density lipoproteins levels. Type specific treatment summary: type 1, that is familial hyperchylomicronemia, treatment fibrate plus diet. Type 2A, that is familial hypercholesterolemia, that is treatment statins. Type 2B, compound hyperlipidemia, statin plus fibrate. And type 3, this beta lipoproteinemia, treatment fibrate. And type 4, familial hypertriglyceridemia, treatment fibrate. And type 5, that is mixed hyperlipidemia, fibrate plus omega 3.
Anyway, this is much better presentation, and you have studied a little bit also that we can make out. Now, if I ask you, what is corneal arcus?
So corneal arcus is a clinical feature of type 2 hyperlipoproteinemia. It basically manifests as a white ring around the cornea as a clinical feature.
Yeah, how do you differentiate between type 2A and type 2B hyperlipoproteinemia?
Type 2B is mixed. There are raised levels of both chylomicrons and triglycerides.
সাইটে যাও
Type 2 hypercholesterolemia, type 2A familial hypercholesterolemia, and type 2B
সাইটে যাও
type 2A
সেকেন্ডে যাও সেকেন্ডে যাও সেকেন্ড সাইটে যাও সেকেন্ড সাইড আচ্ছা আগেই যাচ্ছে আরো যাও আরো আগে
cholesterol.
Type 1 and type 5. 1 and 5.
Hyperlipidemia.
Now you tell 2A and 2B. How do you differentiate?
In type 2A hypercholesterolemia, elevated low density lipoprotein increases.
In type 2A, only cholesterol will be high. In type 2B, cholesterol and TG both will be high. And that will not match with other lipid profile. Which is not high means everything has increased. No. Rest all the parameters will be normal. Except these two, which will be very high. Without any reason.
We can see the plasma. We can see the plasma sample in both the types. In type 2B, there will be slight milkish plasma plus turbid plasma. In type 2A, there will be clear plasma. clear plasma, type 2A.
Very good. Very good. Triglycerides.
Maybe because of
What is extra here? See? TG.
triglycerides, because of triglycerides in the plasma.
Okay. Now, if I ask you the treatment, for the treatment you have prescribed fibrate group of drugs and statin group of drug. Suppose you are having type 2A. Instead of statin, which drug you can give?
We can give Ivolumab, which decreases the LDL levels.
No, statin chara.
Type 2A, we will give statins.
Statin you cannot give. So what else you can give?
Ivolumab, which decreases LDL levels.
Cholestyramine. It was available in USA before, now it is available in India also. Cholesterol drug. Okay, cholestyramine is given. Remember this. I taught this in the class because this has come as question once. Okay, anyway, good preparation and good presentation.
Thank you, sir.
Next group, fast.
Good morning, respected teachers and my dear batchmates. I am Sakshi Umesh Gonda, and today me and my team is going to explain the topic lipoprotein metabolism. I would like to start with the introduction and give a basic idea as to what exactly lipoproteins are. So, lipoproteins, as the name suggests, they are lipids plus proteins. Lipids, they are generally non-polar and amphipathic, and the protein component is an apoprotein. Now, for the transport of lipids, the dietary lipids and the endogenously produced lipids, as they are water insoluble, they form a complex with the protein. It is known as a lipoprotein. Now, so lipoproteins, they are essential for the transport, storage and utilization of lipids. Now, the classification of lipids, we have chylomicrons, we have VLDL, which is very low density lipoprotein. We have LDL, a low density lipoprotein, and HDL, which is high density lipoprotein. Few major components of each of these classes. In case of chylomicrons, we have TAG, that is triacylglycerol and phospholipid. In case of VLDL, we have TAG, triacylglycerol. In case of LDL and IDL, we have cholesterol, and in case of HDL, we have phospholipids and cholesterol. Now, the basic structure of the lipoproteins, the inner core of the lipoprotein, it mainly contains TAG, triacylglycerol and cholesterol esters. Outer to that, we have a monolayer of phospholipids in which a few free cholesterol molecules are also present. There is also the presence of peripheral proteins and few integral proteins. Now, the further topic would be explained by my teammate.
Hello everyone, my name is Imdad Khan. I will be now further continuing into the metabolism of chylomicron.
একবার ব্যাক করো স্লাইডে এই লাস্ট টাইমটা কি করতে লিখেছো ফালতু কপি পেস্ট করে দেখতে পাচ্ছ কপি পেস্ট করে ফল দেখতে পাচ্ছ এটা ব্লক করা যায় যেতে পারতো একটা স্লাইড তৈরি কর সামনে থাকো চলো বলো 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 ঠিক আছে লয়েছো বলো
Chylomicrons have a density less than 0.95 gram per liter. They have 1% protein content and the rest 99% is lipid content. The lipids are mainly triacylglycerol and phospholipids. They are primarily synthesized in small intestine. Their primary role is to transport dietary lipids from small intestine to peripheral tissues such as skeletal muscles, adipose tissues and heart. In small intestine, nascent chylomicrons are produced from dietary phospholipid, cholesterol, cholesterol ester, and triacylglycerol, as well as apolipoprotein B48 and apolipoprotein A. It then enters into circulation through lymphatics. It then undergoes maturation by taking apolipoprotein C2 and apolipoprotein E from coming in from HDL. Then in the mature chylomicrons, the apolipoprotein C2 activates lipoprotein lipase, which is present in the capillary walls of the peripheral tissues. The lipoprotein lipase hydrolyzes the triacylglycerol into free fatty acids and glycerol, which goes into the peripheral tissues and liver. The remaining part is what we have is called chylomicrons remnant, which is taken up by the liver via receptors such as LDL receptors and LDL receptor mediated proteins. LDL receptor related proteins. The hepatic lipase present in the liver also helps in this process. Now coming into the VLDL, IDL and LDL, the VLDL have a density of 0.95 to 1.006 gram per liter. Its major function is to transport endogenously produced lipids from liver to the peripheral tissues. It is produced in liver. In liver, nascent VLDL is produced from phospholipid, cholesterol, cholesterol ester and triacylglycerol as well as apolipoprotein B100. Then it undergoes maturation by taking up apolipoprotein C2 and E coming in from HDL. The apolipoprotein C2 again activates lipoprotein lipase, which hydrolyzes the triacylglycerol into free fatty acid and glycerol, which again goes into the peripheral tissues and liver. The remaining part is called VLDL remnants or IDL. The IDL thus produced has two fates. It either goes into the liver and the liver takes it up by receptor mediated endocytosis, or it goes on to producing LDL by further losing triacylglycerol component and apolipoprotein E. So the LDL thus produced has only cholesterol and apolipoprotein B100. The LDL is taken up by the liver as well as adrenal gland and adipose tissues by receptor mediated endocytosis. A protein called catherine helps in this process. The catherines are present on the inner side of the pits and the LDL receptors are present on the outer side of these pits, which takes up LDLs and binds with LDL and forms LDL-LDL receptor complex. The LDL-LDL receptor complex formed, the catherine then internalizes the pits and the structure produced is called endosomes. The endosome then undergoes shedding of catherine and the LDL receptors are further reutilized. The structure then fuses with lysosomes and endolysosomes are produced. The endolysosomes are then undergoes degradation and the cholesterol is either stored in the tissues or it is utilized for the synthesis of plasma membrane, steroid hormones, bile acids, etc. It further decreases the LDL receptors as well as inhibits the cholesterol synthesis. Thank you, sir.
So next is HDL. HDL stands for high density lipoprotein, is also known as good cholesterol. Good cholesterol because it helps to remove extra cholesterol from peripheral tissue and deliver it to the liver for excretion. Steps of HDL metabolism. First হচ্ছে HDL formation, synthesized by liver and intestine. Nascent HDL, discoidal in shape, rich in apolipoprotein. Second, cholesterol efflux. HDL collects from peripheral tissue. It's a cholesterol efflux.
মাইকে বল, মাইকে কথাটা মাইকে বলতে হয়। আর একটু জোরে বল।
Sir, HDL metabolism. So HDL stands for high density lipoprotein. HDL also called good cholesterol because it helps to release extra cholesterol from peripheral tissue and deliver to liver for excretion. Steps of HDL metabolism. First, HDL formation, synthesis by liver and intestine. Nascent HDL, discoid in shape, rich in apolipoprotein. Second, cholesterol efflux. HDL collects free cholesterol from peripheral tissue and macrophage in the arterial wall. Transport facilitated by ABCA transporter. ATP binding acid transporter 1. Third, esterification of cholesterol. Enzyme acetyl cholesterol acyl transferase converts free cholesterol to cholesterol ester. Convert D-squared HDL to spherical HDL. Fourth, lipid exchange. Cholesterol ester transfer protein convert HDL to VLDL and LDL in exchange for triglyceride. Last, HDL clearance. A scavenger receptor present in liver cell uptake cholesterol ester from HDL without degrading the entire HDL particle. HDL can be recycled by continuing.
Hello. Now we will look upon to the clinical relevance of lipoprotein. The clinical relevance, the lipoprotein classification is like can be called the Frederickson Classification. So in the Frederickson Classification, we got 6 types: 1, 2A, 2B, 1, 2A, 2B, 3, 4, and 5. The one is called as the familial chylomicronemia, where the
In your previous group, they have explained this hyperlipoproteinemia.
Okay.
Can you explain some hypolipoproteinemia?
Hypolipoproteinemia. Has this
Hypolipoproteinemia.
as a hypo when there is no chylomicron. In the absence of chylomicron, the hypo.
Anyone else?
beta lipoproteinemia.
A-beta lipoproteinemia. Familial A-beta lipoproteinemia. কি হয় ওতে বলো?
In beta lipoproteinemia, apolipoprotein B is not produced, so no chylomicron produced, no VLDL and no LDL produced.
HDL.
No VLDL. A beta তে আপনার chylomicrons প্রোডিউস হয় আর A1 deficiency তে
আচ্ছা, what is the difference between B48 and B100?
So the protein content is where 40% and 100%.
কে পারবে? Anyone from the class? বাহ, this is an example of RNA editing. Tissue-wise RNA is transcribed or translated only the first 48 amino acids in the N-terminus that occurs only in the intestine, and all 100 amino acids are translated in liver. So whichever compound is having ApoB 100, they will be taken up by liver, and if they are generated from liver, they will carry ApoB 100. If they are produced from the intestine, they will carry ApoB 48. What is the function of ApoC?
apolipoprotein C activates lipoprotein lipase.
And what is the function of ApoA?
It activates LCAT, that is lecithin cholesterol acyl transferase.
Which one would you prefer to be more in your body?
For more?
ApoA or ApoC?
For what, sir? Can you repeat the question?
Which apoprotein you would prefer to have more in your body? ApoC or ApoA?
Sir, apolipoprotein A1.
Why?
Because it helps HDL to take cholesterol from peripheral tissues to liver.
Okay. Now you said one ALCAT. There is another enzyme called ACAT. What is the significance of ACAT?
It helps in the transport of cholesterol from HDL to LDL and VLDL remnants.
Opposite. ALCAT to opposite. If ACAT is more, that means you have risk of
VLDL and LDL will be higher.
CAD.
Coronary artery disease.
Fine.
না না ওকে বল বলুক শেষ করে না না শেষ করে
টাইম আপ
না না ঠিক আছে আর দুইটা আছে দেখি বলুন না
আরে টাইম আপ
আর কি স্লাইড
আর সাইড আছে
হ্যাঁ
আর সাইড আছে
আর স্লাইড নেই
নেই ঠিক আছে এই গোলাম এরকম ভাবে বললে হবে না নট অ্যাকসেপ্টেড হ্যাঁ আরে হাতে করে লিখে যায় কাগজ দিয়ে করে বল কাগজ থেকে বল সেমিনারে বলার সময় কাগজ থেকে বলতে পারো ঠিক আছে ইট ইজ বেটার টু বারবার ওখান থেকে পিছন ফিরে টাকা হচ্ছে কাগজ থেকে বলাটাও বেটার ঠিক আছে কিন্তু প্রিপেয়ার করে আসো যেমন ওরা প্রিপেয়ার করেছে প্রিপেয়ার করে আসো
খুব ভালো বলেছে আপনারা দুজনে প্রথম দুজন খুবই ভালো বলেছে
ফাইন খুব ভালো বলেছি
আই মাস্ট বিগ আ ফ্যান আমি নিজে হাত